Hey everybody, welcome to webinar 2 of our Microbiomes for All webinar series. I'm Rachel Horak and I'll be your tech support for today. If you need live transcription, you can request that in your Zoom toolbar and below. This recording will be made available to the webinar participants within a day. And if you need any tech support or you want to ask an anonymous question, go ahead and send me a private message to the chat. And at any time, ask any questions about the content that you wish, and our facilitators will make a note of it and address your question when they are ready. Okay. So with that, I'll turn it over to our presenters and have fun.
Thanks, Rachel. Good afternoon, I think, to everyone. I'm going to just share my screen and we're going to get started. Okay. And everyone can still hear me? Okay. So this is the second webinar, as Rachel said, and we're moving on from the getting our FASTQ files loaded into Nephele, and we're going to look at, we spent a lot of time looking at the outputs from those files. And so if you're looking at our little flow chart here, we're in this second position, and we're moving into the kind of the more hardcore data analysis. So just some of you we've heard responses from, and I just wanted to kind of get a sense. I can't see the whole, well, maybe I can see the whole screen. Let me see here. I don't know, I guess I can't. How many people were able to upload FASTQ files to Nephele, if you had a chance to do that over the last week? You can raise your hand or type into the chat. Davita's giving me a thumbs up. I see Stacy. Is that you're raising your hand for a question? Or you were okay? So yeah, I should have just said thumbs up. Anyways, okay. Yeah, right there we go. Oh my gosh. Okay. Great. And so hopefully, if you were able to do that, you got log files back, and we're gonna be getting into those log files more. And so. Let's see. Oops, let me advance to the next slide. All right, Avram, are you, or is this Davita? Is this your slide?
Yes, yeah. This is Davita, but just I'm answering some questions in the chat just to let you all know. We had a technical glitch where someone in the group had downloaded the FASTQ files to their own computer, and so we didn't realize this until Rachel let us know yesterday. So we apologize for that. Files are back in there now. You should be able to access them. So if you can't access the files, let us know. But please, folks, if you are downloading things, make sure that you don't delete it for everybody else if you can. But just to let you know, so this is our Jamboard, and we just wanted to emphasize for everybody that we see there's a lot of folks who are working on the same sample types. So what we're hoping to do is we've put together in your session folder, you will see a resources document. And we're going to add to that any resources that we have that are particular to sample types. So for example, if you're asking us about soil extraction, we're going to put kits in there that we know that you can use for soil and everything else. And then when we leave this session, this webinar, we're going to reach out to you all and see if you all want to work together on things, which would be kind of neat. So we might make a little soil group and a little water group, and then we can collaborate together like that. But that was the purpose of doing this exercise. It wasn't to be a waste of time for y'all, to see what y'all were doing. And we're very intrigued to know about the holy water samples, etc. There's some really interesting ones in here. So we'll be reaching out to you about that too.
I just wanted to add something in here. I see like cell phone swabs, and there were a couple others, but particularly things like if you're swabbing surface, or a lot of times students want to do money. These are kind of low yield samples. There's not a whole lot of bacteria or microorganisms on these samples. So your DNA yields tend to be low. And whenever you have samples with low DNA yields, contaminants are a bigger problem because they make up a larger fraction of the total sample. So just be aware if you're working with some of these low yield samples, you might want to, get in touch with us and we can give you some hints on how to, avoid the contaminants.
And also just to add to that, use a filter, an additional filter when you're doing your extractions. Just run an empty filter effectively. And that's one of the tips that we say to everybody. Always include a filter that doesn't have a sample. And that way you'll know if there's any background contamination in your protocol. So that's a pro tip from last week.
Okay. Am I on the right slide now? Okay. And so here's our pipeline, and here's where your FASTQ files come in. And we're moving now into the QIIME2 and Nephele analysis along some of the other analysis tools. So we'll go ahead and get started. And the first thing that I wanted to, go into was the data2 analysis pipeline. So let me exit out of the slideshow, and I'll kind of try to do things live if the connection is working fine. And if you have questions along the way, Avram and Davida will let me know what those questions are.
I'm still here, by the way.
Oh,
I just took the spotlight off, that's all, because you two are running this. I'll just run the chat.
okay. Okay, everyone can still see the screen, I hope. So here's the Nephele interface, and we talked about the pre-processing last week, and then the analyze step. And we explained how to upload the FASTQ files and the metadata file and to move through with the analyze steps. And if everything went smoothly with that, then you end up with the outputs, which I'll show you just how those look in a minute. All right. So you'll look at a output folders like this, and these came out in my downloads folder. I just renamed them. And from that first run, the single end data2 run, if you were using the demo files that we provided, from that data2 run, you're getting this set of outputs back. And we talked a little bit about the log file and the taxa.biome file. And today we're going to look into the graphs that are some of the outputs that you can start to analyze. And I'm going to open this up. I think the first one that we're going to look at is the rarefaction curve. Let me just switch back to the presentation.
Theo, could I ask you a question just
Yeah.
before we move on? If you have a moment while people are working on things, maybe we should put the output that entire output folder in the drive, make a copy of it.
It is.
Is it in there?
I think I uploaded the whole file.
Let's see.
Is it there?
I don't think so. Is it in resources?
Oh, it might be in resources.
I'll go do that for you. Sorry, apologies.
No problem. So again, here is the outputs file that I just showed you. And the first file that we're going to look at is the rarefaction. And this is a reminder as kind of a resource alert. There are a bunch of statistical tools that get used throughout these analyses. And what I've used a lot just to educate myself and also for the students is StatQuest. And they have a YouTube channel that has pretty much like a 5 to 15 minute video on pretty much all of the different statistical tools that are used, like alpha diversity and beta diversity and so forth. So I'm just going to mention that in case people want to use that if you haven't already discovered StatQuest. There's another one called MathBench, which is also very good. So those are two things to look at. So for the rarefaction curve, what this output tells us is if we sampled deeply enough. So if we have enough sequences in our samples to say that we've covered most of the biodiversity in that sample. And a lot of, an example that a lot of people use is a bag of M&Ms. And so in a bag of M&Ms, you have 5 or 6 different colors. If you were to reach into a bag of M&Ms, you know, if it's in a brown paper bag and you can't see what you're picking, how many M&Ms do you need to randomly pick to make sure that you get all 5 or 6 colors? And so if you pick out 10, chances are you're probably going to have gotten all 5 or 6 colors. And if you pick out 30, you're just going to keep picking those same colors. You're not going to get a new color. If you pick 100, again, you're just spending a lot of time picking, but you're not getting any new colors. And for the sequence data, for these soil samples, it's essentially the same thing. If we look here on the x-axis, we have the depth, or how many reads did we look at. And what the rarefaction curve, what the function does that generates this rarefaction curve is it randomly goes through your data and picks sequences. And so the number of sequences goes across the x-axis, and on the y-axis is how many new novel taxa did you identify? Okay, so how many, essentially the equivalent of colors did you identify? And when this curve begins to plateau, then you know that if you keep on sampling, if you do say 20,000, 30,000, 100,000 reads, you're not going to really get any more diversity. You've already gotten pretty much everything you can get within the first, say, 15,000 reads. And so if you're in this plateau part of the curve, you know that you're pretty good. If you're in the area of the curve that's still rising, then you can say, well, if we sequenced more, if we collected more samples, we could probably get more diversity. So that's the use of the rarefaction curve. The one thing that I'll take a minute to note here is you may have seen when you're looking at these files that in this case, the y-axis is labeled as operational taxonomic units, and you might also see ASVs, which are amplicon sequence variants. They're pretty similar. The newer outputs use ASVs, the older outputs use OTUs, and essentially they're shorthand for the different taxa. And because you have sequences that don't necessarily match to a species that's been identified, you can't match every output to a species. So instead they create these categories, amplicon sequence variants, to represent those taxa, even if they don't match to a species that's in the database. All right, any questions in the chat that I should get to?
One coming from Avram now.
I think you're muted, Avram.
All right. Which one did you tag for me there, Davida?
Missed the part.
Did I miss the part? Hang on. Did I miss the part where we learned what the sequence similarity cutoff is for the OTUs?
Okay. I didn't mention that. And when you're doing the Nephele, when you're using data 2 or Chime 2 through the Nephele pipeline, you're not getting an option to change that sequence similarity. So typically it's set at 99% for species or 97% for genus. And so what that means is that if you have 2 different sequences in your data that are more than, say, 97% identical, then they get collapsed into the same bin. So those 2 sequences get put into the same box and counted as 2. And if you get another sequence that's also within that 97% threshold, that also goes into the same box.
Okay, there's another question. Is there a benefit from Karen Thickman? Is there a benefit to being further right on the curve?
Once you've hit the plateau, there is no real benefit. It's your, essentially, I mean, there isn't a real downside to it either. It just means that you're maybe paying for sequencing that you don't actually need that much. Or, you know, if this is, a lot of this is computationally intensive, so you may not need to work with that many sequences, you could shrink your files and still get the information that you need.
Does it also say something about how many samples you can pool together, perhaps as well, on a flow cell?
You would know more about that than I would. I because I'm not running my own samples. I think that I don't know. It's already after the data. So what do you think?
Well, that's what I was going to say. So folks, if you find that you're not getting all the diversity out of your sample, you have to kind of possibly redesign it and kind of use fewer samples pooled together on the same flow cell. That's what I would see. So if you don't get everything, you need to expand your sequencing, if you know what I mean. Because if you haven't got enough, you have to start over in a way.
And I think I mentioned last week that this demo data set that we've shared with you are the soil samples that we sent to Wright Labs in Pennsylvania to do the sequencing. And you can see here that they're typically getting us between 20 and 30 to 40,000 reads per sample, which in this case is enough to get most of the diversity there. Rachel, is there a question?
I have a question. This participant asked, when I ran my own analysis, no output folder was generated similar to Dr. Muth's folder. What can I do to fix it?
Okay. So what I would say is that we can look at the log file for you and try to figure out where the glitch was. Usually it's with the mapping file. And one of the things that I was going to suggest, and again, this is kind of depending on the flexibility of Rachel's team. If there are like individual questions like that about like, why did my run not work? We can stay afterwards and try to address those. And if it doesn't work to stay afterwards, because I know people have busy schedules, contact us, send us an email. We'll set up a Zoom with you for a time that works, and we'll look at your log file and try to figure out what's going on. But there's a whole, half a dozen things that can commonly go wrong, and it's kind of hard to address those blind without looking at the log file.
Okay, so that person wasn't the only one who had that experience. And the next question is, does it require special software to view the files?
No. So that HTML file output, you can just click on and it opens up. I think I'm using Chrome, but it should open up fine in any browser that you're using. There are other files in there that, if you don't recognize the extension of those, oftentimes, like there's tree files that require a special program if you actually want to see what's in those. There are other text files that you could open up in Excel, but the ones that we'll be working with today are the ones that are the HTML files, and those, if you just click on them, they open up in whatever browser that you're using.
Okay, thanks.
Okay, so that's the rarefaction curves. That's one of the files that comes in the output. And the next file that we'll talk about is the alpha diversity file. But I just wanted to give a little background to the biodiversity measurements that we can look at with the outputs from data 2. I'm just using an example here from Prospect Park in Brooklyn, and I'm kind of giving you the scenario that I use with students to kind of translate that to the microbiome samples. And so I've given two different scenarios. One is that you go through half the park, the north half of the park, and you take 20 minutes on your own to count the number of trees, birds, and animals that you see. And I've shown here that you see 25 species. And then you go through the south half of the park, but you take 2 hours and you go with some friends that are a professional ornithologist and a botanist, and you count what you see. And when you spend 2 hours and you go with experts, now you see 60 species in the south half. And so, would it be safe to say that the biodiversity in the south half of the park is greater than the north half of the park? And the answer is no, because you actually spent more time and you went with kind of better information or better expertise when you were looking at the south half of the park. And so you were able to identify more species. And when you're working with microbiome data files, that kind of length of time translates to how many sequences you're looking at. And the expertise translates to, are you looking with a sophisticated classifier, and are you looking at, say, only at higher taxonomic ranks, like say phylum or class, or are you looking at more fine-grained taxonomic ranks like genus or species? And so, this is kind of an analogy that students might be able to relate to, and it's kind of how I put them in the mindset when we're looking at the alpha diversity for the microbiome analyses. So, Davida, we're going to do a Jamboard.
Yeah, only if the folks want to. So let me just grab that Jamboard link for you.
Stop the share, or
you can keep sharing. It's fine. I'm going to do it right now. So folks, furthering on what Theo just said, we wanted to ask you, given that you had told us what types of samples you were looking at, if you can go to the Jamboard link, it's in your chat right there. Actually, do you know something? Let's share it, and I'll just show people where to go. Let me share there. I'll just share right now. So people know what we're talking about. So in essence, because this is an ASM workshop, we're trying to ensure that we connect everything to the curriculum, right? So we wanted to ask you, if you're looking at your own samples, given the example that Theo just gave you there with respect to Prospect Park, how might you ask questions about diversity for your own students with the samples that you're interested in, be it frog skin, lizard skin, whatever it is, chicken, respiratory tract? What kind of questions might you ask? So here are some examples of things that kind of prompts to help you get started. If you could even post just one question that you might ask your students with respect to your samples, it would help us consider experimental design as well. Blue are the examples of questions, and yellow is the prompt. Okay. Excellent. I see some questions coming. That's great. Everybody do just one. This is great.
TR, there was a question about the rarefaction plot in session 2 working folder. Was it on the Google Drive?
Say it again.
Caleb Fisher was looking for the rarefaction plot in the session 2 working folder. Is it on the Google Drive?
If it isn't, we'll put it there. I think that may have been one of the folders that mistakenly got removed, but it had been there, and we can, if it's, yeah. So I'm seeing here, oops. Yeah, if someone clicks on graphs, it should be there. Davida, are you?
I'm in there looking now.
I thought I saw it. If you went back to the original folder that you were in, the outputs ones, yeah, here.
It's in, yeah, it's in the drive.
Yeah.
Can you do you want to share it? Just show them exactly, point it to them. Just show them how to drive to it. I'll go back to the.
Oops.
So if you're in session 2.
You want me to share, or you want to share?
Yeah, you show them how to get to it exactly, so they don't get lost.
So where do you want me to show it?
Show from the session working folder. So just go into the drive and show them the folder, so they know where it is.
Okay.
So
here's, can people see everything?
Yeah. So soil microbiome data files, I put everything in there.
And we were looking at
curve right there.
Where is it?
Down, 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 down.
Well, I can't see it because something else is blocking. Yeah.
Row curve down the
Right
bottom.
there at the bottom, right.
HTML.
So if you all see that, so we put all the outputs in the one folder, and if you just click on graphs, it'll bring it to it. Caleb.
Okay, so lots of interesting things coming up on the Jamboard. I think we'll kind of curate that and share it afterwards. Oops. So the next file I think that makes sense to look at after rarefaction is the alpha diversity file, because that's what we were just looking at when we're talking about, what can we detect when we're going through the park. And I should be able to get that right from here. If I can find the right. Is it not there? Oh, there it is. So this is the alpha diversity HTML. Oops. I don't think it liked what I just did. All right, so I'm going to open it from my other.
Open it from the other folder. I wonder, is it because I copied it? Maybe that I copied it in.
No. Okay, can people see my screen? And are people seeing the species diversity graphs?
Yes.
Great. So when you open up that alpha diversity, or it's called alphadiv.html, that's one of the outputs from the data2 Nephele pipeline. It automatically generates a bunch of graphs. Actually, a whole bunch. And you'll see that there are two types. There are the Shannon's diversity and the Chow-1 diversity. So those are always the y-axes on all the graphs. And then based on what your metadata file was, it's creating x-axes to match up with your metadata. So you can see the alpha diversity under the different categories of metadata you have. So here's, I think maybe one of the interesting ones to look at is the source. And what the alpha diversity is telling you is this is the diversity within a sample. And it allows you to kind of compare, which samples you have that are high diversity. So here, these soil samples from the Brooklyn College campus quad are pretty high diversity, whereas say the Lily Pond sediment, which is also on campus, that's a relatively low diversity sample. When we talk about, so I'm going to switch back to the slideshow. So when we talk about these metrics of alpha diversity, there's a few things that you can ask for your students to be able to do. And one is, like I just did, to identify those samples that have the most or the least diversity, which is essentially asking them to read the information from the graph. Another is to assess whether or not differences are likely to be significant. So we have some graphs that have like the box and whisker plots. So you can use that as an opportunity to talk about what's in the box, what's in the whiskers, where's the mean, and to kind of say, these show up at a different place on the graph, but they're overlapping. So what do we say about the significance there? And then one of the last things that's kind of the fun thing to do is to hypothesize about, well, why is my lily pond sediment so low diversity and my houseplant soil is high diversity? And so then you can start to speculate why that might be. That also generates questions that you can use other data that come from the outputs to start to answer those questions. So I think there's a lot of distance that you can get out of these alpha diversity plots. The other thing that I wanted to point out about the alpha diversity plots is that there are some, so in the case that you get from the Nephele output, for the alpha div file. You have chow1, which is just a measurement of richness. Okay, oftentimes you can also see like a species count or observation counts. That's just counting the number of different species that you saw. And so it's just looking at what we call richness. Other metrics like the Shannon's diversity or Simpson's diversity look at evenness as well. And so I've used this as an example here on the left of that, walking through Prospect Park and seeing what you are able to identify. And in both cases, the north half of the park and the south half of the park, a total of 12 species were identified. And you can see that I put in total observations, they're both around 90. So the intensity of looking was the same. The total number of species or the richness was the same. But we want to talk about evenness. And if you look at the north half of the park, over 80% of the sample or over 80% of the observations come from a few species like maple trees, starlings, house sparrows, and humans. So a lot of that diversity or a lot of what you see is really in just those 4 species. Whereas if you look at the south half of the park, there was more evenness where there weren't just a few species that were making up most of the observations. And so when you're looking at a graph that with the Shannon's diversity, that evenness is taken into account. And so that gives you kind of a better picture of what the alpha diversity is for that sample. And one of the other things that I think is nice to do with these data, and again, this is a good starting point for students, is to generate a rank abundance curve. And what the rank abundance curve does, I've got the example from the data over here on the left in this graph on the right, is that you're sorting the data by the most highly observed species all the way down to the most infrequently observed species. And you're listing those species along the x-axis and the number of counts on the y-axis. And so this concept of evenness, you can kind of see illustrated here. This, I tend to tell you that I'm colorblind and I can't tell what color these are, but this front one here is of some color.
A lighter green color. The one at the back is a darker green color.
Okay. So the lighter green is from that north sample. And you can see it drops off really quickly. Like most of these counts come in those 4 species that I mentioned. And so you get this steep drop off. Whereas you have a more even curve with the south side where, yes, those same species are the most common, but the other ones, aren't so much different. And so making this rank abundance curve, I think, is a good starting point. And again, with an example like this, it's pretty easy to conceptualize. In the next slide, this is what our data look like when you make a rank abundance curve. And I've done this at the level of phyla. And so you can see all of the phyla in the sample listed across the x-axis. And you can see the most abundant ones on the left and the least abundant ones on the right, and the counts, or how many times each one of those phyla showed up in these samples that I'm showing. And I just chose to make, I just chose 3 of our samples, one of the peninsula soils, one of the flower bed soils, and one of the houseplant soils. And if you look at the left, you can see that they start at different levels of abundance for their most abundant species, this is phyla. But they show that same pattern of a rapid drop off and then this long tail of diversity. And this is typical for most microbiomes and definitely typical of soil microbiomes. The soil microbiomes are really dominated by actinobacteria, acidobacteria, proteobacteria. We see that in all the soils that we look at. And again, so maybe 80 to 90% of the reads, again, depending on the sample, may just fall into those top 4 or 5 phyla. These others are there, but they're very uncommon. And so that's something that I think is worth kind of getting, kind of making students aware of, because first you get all excited, like, oh, wow, everything is like, there's so much diversity there. But then you realize that a lot of that diversity is really quite uncommon. So I'll pause for a sec. Are there questions in the, oh, I should say that there's a video for making this Excel graph. I think that's in the, is it in the resources folder or is it in the session 2 folder?
I'm going to double check right now.
I think it's if your students, again, the students that we work with at Brooklyn College, a lot of them don't have experience with Excel. So making a graph like this is kind of a good entry point. Avram, are there questions from the chat?
No, not at the moment.
Anyone want to just pipe up and ask a question?
I had a quick question. You had mentioned that asking students to think about like whether or not the different alpha diversities might be significantly different from each other. And I was wondering if there were any like statistical tests that are in this software somewhere to allow them to actually test it, or if there's like an easy test that students can do.
So other than making, those interpretations, oops, let me see if I can, when you're looking at, in this case, this Shannon diversity plot on the right, you can, make those assessments visually by looking at to see if there's overlap or not. The outputs in Nephele don't do what you're asking for, but that resource that I mentioned earlier on, StatsQuest, you can go into that and they have lists of, the t-tests and chi-squared tests, and they go through the tests that might be appropriate for addressing questions like that. I should say that in our intro micro class where we use this is, one element of what we're doing, and we're not getting into a lot of the statistical analyses, so we don't cover that. It's a shame, but we just, we don't have enough time. There is a semi-biostatistics course that students can take, but it's not required. Does that answer your question? Okay.
So Karen Thickman asks about contaminants and low ones. Are they low abundance ones?
Oh,
contaminants or not? How can you tell the difference?
Yeah. So excellent question. And if you watch the video, I mentioned this a little bit, but notice here that firmicutes is high in the houseplant soil, and it's not really very high in any of the other soils. And if you look at all the data together, and I'm only just showing these 3 samples, you'll see that those 2 houseplant samples are enriched in firmicutes. And so that includes a lot of the bacteria that you find growing on human skin. And this is kind of one of the fun speculation things, is that, well, this houseplant is actually in one of the biolabs. So it's just getting all this dust and air and skin sloughing off of people. And so, we speculate, well, maybe that's why there's more firmicutes in those samples than, say, from the park or from the quad. It's hard to tell if it's, really a contaminant or not, because that's, really a part of the soil, but it would also be easy enough to come in off of your skin. If you look at, I think we'll see it maybe in the bar plots if we get to that today. There is a negative control sample, and it's just loaded with staph, I think it is. And that's probably because when I was prepping this, as Davida was mentioning, we just run nothing through one of the columns. So that's our negative control. And probably I inadvertently got, some skin stuff in there. And so because it's such a little, since that's the only game in town for the PCR to amplify, it just amplifies that contaminant through the roof. And so that showed up as like more than 80%, I think, of that particular sample. And so in a case like that, where that's a negative control, well, you can be pretty sure that that's a contaminant. But from the houseplant soil, it's harder to say, because yes, it could have been a contaminant, or, maybe it wasn't. So I'm not sure if I'm answering your question, but you can usually try to figure it out.
Yeah, so that was me asking the question. So really what I meant was not so much those ones sort of on the left side, but the ones way down on the right. right? So those ones down there, like, how do you know when you're, I know that from the rarefaction curve, you can say that you've reached, saturation, but how do you know that those ones down there aren't just, random sequences or sequences that are misidentified if all you're seeing is a few of them, right? So
Really,
yeah, I guess that's, how do you know that those are real?
Well, maybe I could answer that because the first question is it reproducible? The second is, do you have, if you're controlling for all the equipment and the tubes you're using with a no sample control, it shouldn't come up. But I'd say, if it's on the far right of the curve and it's reproducibly found but isn't in your no sample control, then I would say that's probably a result.
yeah, that's exactly.
Even if it's lower, even if it's really low abundance.
That's what I was going to say as well. And partially the reason why we don't do more than one sample when we're doing this is just cost. But ideally, you should be taking more than one sample and showing the reproducibility of what you're finding.
Yeah, and actually, like I said, there are replicates for these samples. When I was doing this rank abundance, I only did these three just, so it wouldn't take me a half an hour to make the video.
So would you make each of these, when you have replicates, would you make each one separate like line, or would you average them, or like how do you deal with the replicates in that context?
Right. So I think with the rank abundance, you're trying to show this diversity, how it drops off quickly. For looking for contaminants, I wouldn't use, this rank abundance plot. I mean, to answer your question, you could put all your samples and then you'd go in the Z dimension, you just have them all lined up and you might be able to, see some of it there, but that is easier to detect in a bar plot or some of the other analyses.
Okay.
But it's a good question. Any other questions out there? Okay. Right, so there is a video. It's a little bit rough, but maybe people can get something out of it. The final thing that I wanted to mention about alpha diversity is that this doesn't show up in the data 2 outputs from the Nephele pipeline, but it does show up in the QIIME 2 outputs, is alpha diversity measurements that take into consideration phylogenetic distance. And the one that shows up in the QIIME 2 outputs is Faith's PD, or Faith's phylogenetic distance. And the example again I give the students is that, if we go to the Bronx Zoo and one person goes to the aviary and they count 10 different birds, one of each species, and someone goes to the Madagascar exhibit and counts 10 different species and one of each species, then you have two samples where you have the same richness, 10 species, you have the same evenness, one of each. And so you would say, oh, on Shannon's diversity or Simpson's diversity, those are going to score identical because the richness and the evenness are identical. But obviously, if you're going to the aviary, all of those 10 species are from birds. And if you go to the Madagascar exhibit, you could have reptiles and plants and so on and so forth. So if you measure those distances by some tree branch on a tree, you can see that the Madagascar samples are more diverse. And so that's a third element into these alpha diversity measurements that's important to keep in mind, is that phylogenetic distance. Okay, so I'm going to now move to one of the other outputs, and these, I'm gonna look at the principal coordinate analysis, and there's a couple different options, but I'm gonna look at the Bray-Curtis principal coordinate analysis, which is one of the other files that comes in the data 2 outputs.
There seems to be an issue coming up about these HTML files.
We'll have to fix it, folks. We copied them into the drive and I think it messed up with the formatting, so we'll have to figure it out. I've tried downloading them and opened them from a download and it doesn't seem to work. So we'll address it and we'll get back to you on that. Okay. It's because we had to upload them in a hurry. So we'll figure it out.
Yeah, the ones that you're seeing, like, can people see my screen here?
Yeah.
We can still see your screen. We're just going to need to help them with the files.
So what you're looking at here, this is the output that I generated locally, on my computer. This was the output file that Nephele gave to me. So if you were able to load your FASTQ files and you were able to get an output folder that has these files in it, then all I'm doing is double clicking that and they're popping right up. So as Davida said, it looks like it's something from the transition to our Google Drive, and you'd be downloading that something gets messed up. But if you were able to do this and get your own output files, then it should be working fine from there. Right, so this is the PCOA plot, and I will switch back to the slide presentation for a moment. And there are two different plots that come out from the data2 pipeline, and that's the Bray-Curtis, which I'm showing, and the binomial. The difference between the two is that the Bray-Curtis is weighting the samples. So that means that the abundance is taken into consideration. So if there's a sample, or sorry, if there's a taxa that has a high abundance, that high abundance is factored into how these samples are clustered. Whereas the binomial is not looking at any of the abundance information, it's just looking at the different taxa that are showing up in the sample. And let me just go back to here. Okay, and there is a pretty good description of how these PCOA plots are generated on StatsQuest. Essentially, these are beta diversity measurements that are looking now across samples and comparing samples to one another. The alpha diversity metrics were the diversity within a sample, and the beta diversity metrics like the PCOA plot are telling you which samples have a similar community to one another and which ones are more different. So what we look for on these plots are clustering. So samples that cluster are more closely related than samples that are distant from one another, okay? And when you, in this HTML file that you open, you can click on the dots and it gives you some of the information about what that sample is. And you can see that there's a cluster over here on the right-hand side. And when I'm kind of mousing over these are all soil samples. And there's another cluster over here in the upper left-hand corner, and these are the lake and the pond samples. And I can't, hopefully you can see it. I have too many things open. Over here in this lower right-hand corner, there's a cluster of 3 that are all from the leaf litter samples. And so what the PCOA plot really does a nice job of is saying that the microbial communities, or in this case, because we're doing 16S sequencing, the bacteria and the archaea in these samples, the community from the soil are quite similar, and they're all clustering together. So there are elements, there are members of this soil microbiome community that are all similar, even though they're, some are from the campus quad and some of them are from the park that's, a few kilometers away. They all tend to be quite similar to one another, as opposed to the sediment samples that were taken from a lake and the lily pond nearby, and also versus the leaf litter samples. And I think that's really cool to be able to see that these communities are quite different. And the leaf litter samples are actually just taken right above those Prospect Park soil samples. So they're literally, just one layer above those soil samples, but the communities are incredibly different, or at least different enough to really visualize that on a plot like this. And so this is when, if you're able to collect enough samples that you're looking at, some from soil, some from water, or if you're looking at, some from one type of insect and one from another type of insect, you might expect to see those clusters form among those samples. Okay. Are there questions about the PCOA plots? They're, like I said, the Bray-Curtis and the binomial, they'll look very similar. They're just generated from kind of a slightly different use of the data. Were there any questions in the chat that came up? Anyone want to pipe up and ask a question?
There's been some issues with opening the files, but I think we're good. All right, so no, Karen asks, will it make one for each column in the meta table file, like for alpha diversity?
Wait, can you read the question again?
Will it make one for each column in the metadata file, like for alpha diversity?
No, so it's not in these plots that come out of the data2 pipeline. You're limited to how they're grouping them by pond or soil and so forth. Similar plots, and I don't think we'll have time to get to it today, but you also get PCOA plots out of the QIIME2 pipeline. They're also very nice. They're also three-dimensional, which is kind of cool, or they have 3 dimensions, so you can go in that Z plane. And in there, you have the ability to kind of change the sample groupings. And if we don't get to it today, then I can show you in the 3rd session. But yes, there is an option to do that for the output from the QIIME2 pipeline.
So we only have about 5 minutes left, Theo, just so you know, we're running really short on time.
I got my timer here.
Good man.
Okay. So that is, those are the outputs from the data2 pipeline. So if you went into Nephele and you chose analyze and you selected data2, those are the files that are in that graph folder that you can begin working with. If you also ran the QIIME2 pipeline, you get, again, like I said, some of the similar files, although they tend to look a little bit different because it's coming through the QIIME2 pipeline, and you would view these on QIIME2 view. And so here you have, this is the output from the QIIME2 pathway, sorry, the QIIME2 pipeline, and there's a whole bunch of files. The ones that end in QZV are ones that you can visualize looking at the QIIME2 view, and I'll show you how that works in just a minute. So let me change my window a little bit. So if you just Google QIIME2 view, it takes you to this interface, and you can drag and drop, or you can click the files that you want. And so I'm just going to click, and here are those lists of files that I can open. And it's you can also open the QZA files, but it's just text. So if you want to actually see something there, you want to look at the QZV files. And the first 3 files that I'll just talk about summarize the data. So there's a table QZV, a taxonomy QZV, and a rep sequences QZV. The table QZV file gives you some general information about the samples. And if you click on the tab for interactive sample detail, it lists your samples and then this feature count. And so these feature counts are equivalent to those ASVs that I was mentioning a few moments ago. So there's some general interesting information just about the samples themselves in that table QZV file. If you open up, and I'm just going back to that QIIME2 view, and clicking on click here to open. If you look at the taxonomy QZV file, here are all of the taxa that showed up in your samples. Okay, so here you're getting the how the QIIME2 was able to match your sequence to a given taxa, and all the information is provided here. And you have specific feature IDs in this first column. And you can download this information as a TSV file that you can open up in Excel. And then the last of those three is the RepSec, the representative sequence QZV. And if I open that up, here I can see the sequences that were actually used for each of those features that I was just showing you in the taxa file. And if you click on these sequences, it'll take you to a NCBI blast interface where you can blast these sequences to find out exactly what the hit was. You can also do that, and what I usually do is I usually download this table and the taxa table, and I merge the two so that I have my taxa with the sequence that was associated with it. And then students can go ahead and blast those individually if they want. Okay. So I know that we're kind of running short on time, and I don't want to kind of rush through the bar plots, so we can hold that for the third session. And I'm just going to jump ahead. So we left the third session. We didn't program that much stuff for the third session because we wanted to be able to kind of respond to what you guys were interested in us focusing on. And so I will probably talk about the bar plots, and I'll also talk about getting into Microbiome DB, which offers another set of tools. But we wanted to ask you, what things would you like to cover? We can cover heat maps, we can cover Venn diagrams, there's all sorts of options. And Davida, was there a form for this, or how are we gonna record, get this information?
So what we've done is we've basically, we've made another Jamboard. So if you go to the Jamboard, if you can give me share screen again there, Theo. Let me go over here. If you go up to Jamboard, remember where you were just a second ago, you're asking your questions there. If you click just right here, you'll see we've asked you, what would you like to see covered in session 3? So if we're asking you to do this as quickly as you can, if there's something that you really, really want to see us do, put it on there and we'll try and tailor the workshop, the third workshop, to your needs. In addition, questions, if you have any questions on session 1, I've reopened that folder, that file for you, so you can go in there and ask your questions. I'm going to set up a question document for session 2 right now. It'll be in the drive, so you can just go there directly and add your questions there. We will try to answer your questions as they come. So one last question was with respect to Faith PD. That was the last question I saw added in the chat. Theo, did you generate an output for Faith PD that someone could find in their file?
So the Faith PD is one of the outputs from QIIME2. So let me share my screen for a second. Oops, that's not what I wanted to share. If you are in the output file for the QIIME2 pipeline from the Nephele, you have a alpha rarefaction QZV. And if you open that, here is the option for looking at the alpha diversity with the FASTQ metric. And they also have Shannon's. And then the observed features is just a richness count. So you have observed features, that's simply richness. You have Shannon's, which is a combination of richness and evenness. And then you have FASTQ, which includes the phylogenetic distance. But it's in this representation, it's not the same. You saw, like, we had the barn whisker plots for the alpha diversity outputs from the data2 pipeline. Here you need to download as a CSV file and then replot if you want those to look, if you want to be able to compare them directly to what the data2 has, or if you want to have more flexibility. So if you just click on the CSV, you'll have an opportunity to get the data that you can graph in any way you want. Just one other thing, you can, all of these things you can explore and you kind of learn by playing around. And I'm always saying, like, oh, I didn't know you could do that. You didn't know you could do that. So you can click on these and add them back one at a time if you want, if you're interested in looking at just specific.
We would recommend that you kind of explore. So for next week, people are asking what their homework should be. Ask us those questions, as we said, help us plan session 3, and then explore, try and generate some plots, just see if you can get through any of it at all. That's what we'd ask for you for next day. And then one final question from Anne Est. Is there a way to know what parameters Nephele uses for data2 and QIIME?
Yeah, so I haven't been opening those files. But if you look, when you look at the QIIME2, there's usually a tab, sorry, when you're looking at QIIME2 view, there's a tab for details and provenance, and that tells you exactly what's going on. And if you look at the log files in the data2 pipeline, that also tells you what's going on. And when we were in the, let me just, when it's kind of going to be hard for me to show it without actually running through everything, but when you are doing, say, analyze or pre-processing or explore, there's always a tab that says, more details, or I can't remember always how they call it. And in there, it tells you kind of what the parameters are. But the thing, so the nice thing about Nephele is it's, graphical user interface. So it's pretty much clicking. But in order to make it, nice and clickable, they don't give you the flexibility to alter all the parameters. Now, if you go and run QIIME2 through the command line, then you can actually tweak every single parameter. But the trade-off for having the ease of use with Nephele is that there are some parameters that you can change, but you don't have access to all the different parameters. That makes sense.
That's kind of what I'm wondering, is how much of this could I just push through versus go, need to go to the command line to tweak stuff. So thank you very much for that. I appreciate it.
Yeah, I would again, say that what I've shown you is what I do with students in the classroom. And the objective is teaching them about microbiomes and microbial diversity and so forth. It's not a bioinformatics class. It's not a statistics class. And it's an intro level. So I'm not going into those fine-grained details. I'm also not intending to publish this. I mean, at most, these students, there's a science fair at Brooklyn College. These students might present their project at Science Day. Now, that's a pretty low bar in terms of the data that they would need to present. And this is all fine for that. If you want to go to a national conference, if you want to publish, then you're probably going to want to make sure you can still use Nephele. The data are good. It's just that you probably want to pay more attention to those little details to make sure that it's doing what you think it's doing.
And another question from Anne is that how would you organize this in terms of your class time?
In terms of class time? Right.
A different Anne.
Oh. So
Marianne.
like I said, this we do in a lab, and the lab is about 3.5 hours. And so we have a couple of the labs throughout, it's a 14-week semester. So we'll have a couple of labs where I'll take an hour and a half or so in a lab section to basically kind of walk through all these steps in the classroom. And I'm doing it with a big screens in the lab, and they can see everything that I'm doing. No question about it, the best way to do this is in a workshop where everyone has their own laptop and we're all kind of floating around all the problems that you're hitting with. Why is my metadata file not uploading? Why are my FASTQ files not loading? We can come over and look at that log file with you. In this context, kind of we're about 60 minutes and I can't look at your computer or your log file, so it's a little bit hard. But in the teaching format, I can do that if students bring in their files and I can look at it. We have iPads that they can use and open files on and stuff. So that's the way. But in terms of time, our microbiome lab is spread across 4 weeks, but it's not the whole session for each one of those 4 weeks. It's a modular implementation.
And we started doing this because of the pandemic. Just so you all know. So originally, the program used a lot of Excel and different tools that were kind of more accessible to students. And we have a bunch of resources on the CUBE site. So I wanted to just point this out to you. So we put our resources folder on there. We're adding to it. We'll highlight any additional resources in addition to Nephele that you could use. But remember that part of the reason why we started using Nephele was because of the pandemic and because faculty couldn't do what they were doing before with students. So for many of us, we focus on the DNA extraction, we focus on the micro, and then we do a little bit of analysis. But the pandemic has shown us that sometimes in some situations, you might have to do more analysis with students. So our goal is to make Nephele student friendly. So it's going to take time, but we're figuring out the kinks as we go.
Yeah, it's this kind of stuff is great if you're doing a distance learning or remote stuff, because you can just sit on the computer and work through everything. So that's what I've been doing with the students in my lab through Zoom. It works really nicely.
Put the students in groups and they can figure things out together, that kind of thing,
Other questions out there?
My students want to present on Research Day and Campus. That's great. It's awesome. We want them all to present if we can. It's great. Okay. So y'all know what your homework is. Try and do something if you can. Don't feel pressure, but do ask questions and do try and make suggestions as to what you want us to do for next week. The earlier you do that, the better for us, because we are also teaching. And we have to try and get things done in the evenings. It was lovely to see y'all.
With that, I'll remind everybody, take our post-webinar survey, please. These facilitators greatly appreciate any feedback that you have. It'll take you literally a minute, click, 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 and let us know how we're doing. Okay. So I'll stop the recording.